Hi class. What we're going to look at now are the different components of DNA, so you can see them up here on the left. And then here in a little bit, we'll actually look at how we put them all together and make our DNA structures, and in particular the alpha helix. All right, so at the very beginning, when we look at DNA, it's got three main components. It's got a phosphate group, so that's here in purple, right? So it's got a phosphorus, and then it's oxygen. So it almost kind of looks like phosphoric acid that we've talked about previously, except that it's bound to the sugar. All right, now we do have a sugar. It has kind of this ribose unit here, and the reason we call it deoxy is it's missing one extra oxygen. All right, so instead of this having an extra alcohol, so it's deoxyribose, missing this one. All right, so these two will always be the same. All right, so every DNA unit is going to have a phosphate, a sugar, and that's going to make our phosphate backbone here in a little bit that I'll talk about in the image on the right. All right, the part that does change is this base. So we tend to call it a base, but it's actually a nitrogen aromatic compound. And this basically it has nitrogens which can pick up protons, but we'll call this one of our bases. In DNA, there's 4 of these. Right? And so if you look over to the right briefly, we have adenine, thymine, guanine, and cytosine. These are our 4 bases, and everything else, the purple and blue ones over here on our left, are going to stay exactly the same. Just the base is going to switch out. All right, so let's take a second and let's look over on the right. So if you look at this structure over here on the right, first start on the left. So up here on the top where it says 5' end, right? So the dash means the prime. If you notice, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. That's always going to be the same. So we're going to essentially make a polymer chain alternating between that phosphate and sugar all the way up and down. And if you look on the right, it's the exact same thing, phosphate, sugar, phosphate, sugar. The only thing that changes is the base that we use. Right? And so that's what makes this work so well is on the left, the phosphate and sugar are constantly going to get attached. Now, what this 5 and 3 are talking about is where they're attached. So if you look at this image right in the middle, this is telling you where the 5' is. So it's the CH2OH, and then the 3' is this OH down at the bottom. When you come back to the picture on the right, I'm going to start at this top one where the red mark is. This is my CH2OH that we saw over here. And so that's why it's telling you that's on the 5' end, because it's just literally telling you where does this phosphate attach. The other phosphate is going to attach to the 3', right? It's the same over here. And so what we're just talking about is the sugar is always going to attach in 2 positions, right? It's going to attach to one phosphate at the 5'. It's going to attach to the second one at the 3'. Right? And then the base pair will always come off here on the right. Now, when we do this, it works really, really well because these just alternate and we just plug and play the bases that we want. All right, now, these bases are going to specifically match up. So if you look here, and I'm going to use, let's go blue. These dotted lines are highlighting hydrogen bonding. All right, so when bases match up, they're very, very specific. So one of the things you guys might have heard before is that we always get AT pairing and we get GC. All right, and the reason we get this is because they have to match specific hydrogen bond donors. So here our NH2 is the hydrogen bond donor. The oxygen of the carbonyl is the hydrogen bond acceptor. And so they line up perfectly to form these hydrogen bonding interactions. And so they stick together. GC actually makes 3 of them. So that's why it's a little bit stronger interaction. Right? And the same, we're matching up the pairs. If you try to mismatch them, they just don't form good interactions. And specifically, sometimes they form bad interactions. Like if you're trying to put 2 things with negative charges, push them together, they're similar to trying to push a magnet together with the same polarity, right? They repel each other. Once you flip them, they match up really nice. All right, so what this lets us do is if we knew one side of this DNA, so one strand, we could automatically know the other half because everywhere there's an A, there'll be a T on the other side. All right, same thing here. Everywhere there's a G, there'll be a C on the other side. Same idea up here. So this purple represents another G, right, a guanine. So that means on the other side, we automatically know it has to be a C. And so that's why you always see this green and pink, and these are just colors picked out for the example to highlight those specific base pairs, but you're always going to see the A and T matchup, T and A matchup, G and C, C and G. All right? And so as long as you know one side, you can always solve for the other. And that's really, really important because that's how we can make copies of DNA, right? We can take one strand and match up the other, and so it lets us make copies of the DNA. All right, so let me just highlight a couple things. A couple things you should keep in mind is that DNA is always made up of 3 components: phosphate, sugar, and a base. Right? And the base is either going to be one of these 4: the adenine, thymine, guanine, cytosine. All right, so one of those 4 will always be our base. The phosphate and the sugar won't change. The phosphate and the sugar are going to make this phosphate deoxyribose backbone, right? Because it's going to constantly alternate phosphate, sugar, phosphate, sugar. Right? And then the same on the other side. The phosphates are always going to be connected on the 5' position and the 3' position of the ribose. Right? And the deoxy just means we're missing an oxygen. Right? Deoxy, we're missing that oxygen. All right, and then lastly, that the bases in the DNA, they're always on the inside, pair up really nicely because of hydrogen bonding. All right, you're matching up donors, hydrogen bond donors, and hydrogen bond acceptors. And so it always makes them specifically pair up. So it's always going to be AT and GC. All right, last thing about the overall structure is if you notice, these phosphates are charged, and so they point to the outside. So they interact really well with water, right? So water can interact really well with things that are charged. These bases tend to be more nonpolar, and so they're actually buried inside of the DNA. And so you get this nice hydrophobic pocket that these bases can sit in. And so all of these lead to really good thermodynamic features that they're going to make this form really well. And so this is why that alpha helix structure with the double strands works so well, is because we put the polar groups on the outside and the nonpolar groups in the inside. Plus, we form these really nice hydrogen bonding interactions, which are good interactions that help stabilize the molecule. All right, class, I hope that helps.